EBSCOR, the experimental program to stimulate competitive research, is a National Science Foundation grant program. The goal of EBSCOR is to strengthen science and engineering research and education throughout the United States, and to enable states like Wyoming to be nationally competitive in these areas. EBSCOR aims to improve scientific infrastructure in Wyoming and build science, technology, engineering, and math research capacity across the state. One way Wyoming EBSCOR helps strengthen science research and education is through student programs such as the Summer Research Apprentice Program, also known as SRAP. SRAP is an intensive 6-week paid research experience for high school sophomores, juniors, and seniors aiming to attend college. It offers students a hands-on research opportunity in science labs across the University of Wyoming campus. Students work one-on-one with a faculty mentor throughout their time in the program. SRAP not only provides students with a paid internship and real research experience, but also helps prepare them for college life.
Hello, my name is Danielle Obando, and I studied in the Department of Pharmacy with Dr. Lina and Dr. K. The title of my project is Cathepsin K Knockout Attenuates Starvation-Induced Liver Damage in Mice. As an introduction, we focus greatly on starvation, which is severe malnutrition. It is currently one of the greatest threats to today's public health, maybe not so in the United States, but greatly in third world countries such as Africa. The importance of starvation in this project is that it induces autophagy. We focus on the liver in mice. It is a key organ which is necessary to live. It regulates chemical levels in the blood, carries waste to small intestine, and produces proteins, as well as regulating blood clotting. Autophagy is a catabolic mechanism in which unnecessary or damaged cell parts are degraded in the lysosome. It can either be beneficial to the organism or it can also cause the cells to die, really depending on the circumstances. It has 3 main roles in the liver. It balances nutrients and energy and metabolism. It clears misfolded proteins in liver disease, and it participates in organelle integration. In our study, we used 2 sets of mice. Half of them were starved. Our starved mice were starved for 65 weeks, so in this case, it was considered a prolonged starvation in which autophagy was negative for the mice and caused more liver damage. This is, like this one, a basic diagram showing the process of autophagy. You have the cell parts surrounded by a double membrane. The double membrane is a marker of autophagy. It then turns into the autophagosome, which combines with the lysosome, in which it loses one membrane and becomes the autolysosome. In the autolysosome, the parts are degraded. This is also the same, except here we highlight 3 of the markers that we use in our study. When autophagy is activated, these markers are also activated. As I said before, autophagy in this situation would be negative for the cell, and thus in our experiment we tried to decrease the amount of autophagy. Cathepsin K is a cysteine protease which we use in order to help decrease autophagy. It is prevalent in certain diseases such as bone diseases, mainly as arthritis, osteoclast function, and pinocytosis. In studies of arthritis, they compared the levels of cathepsin K in normal patients without arthritis and in patients who did have arthritis. They found that in patients with arthritis, there was higher levels of cathepsin K, and thus an inhibitor could be targeted in order to decrease the cathepsin K levels and also decrease the amount of disease. The reason why we picked cathepsin K in our study was because since we're tracking autophagy, we needed something that was related to that, and other members of the cathepsin family have been shown to be participants in autophagy. Also, our pharmacy department in UW has done research on obesity-associated cardiac disease and cardiac hypertrophy. The purpose of the study was to determine the role of cathepsin K in knockout starvation-induced cardiac dysfunction, focusing on autophagy. hypothesis consisted of the first being cathepsin K knockout would rescue starvation-induced morphological changes and lipid accumulation in liver, and also the knockout would help to inhibit starvation-induced autophagy in the liver. For materials, we used 2 sets of mice. The first set was fed a normal diet, while the other was starved for 65 hours. Within those 2 groups, we had 2 subgroups. The first group was normal regular mice, the second had a cathepsin K knockout. We used several antibodies and 2 dyes for our methods. The first method was oil red O staining, which allowed us to detect liver accumulation within the cells. We used a SRAP to cut frozen liver samples into 5 micrometers, and then we stained them using the process and observed them under a microscope. For the H&E staining, we used also 5 micrometer sections. Instead, we used the H&E staining method, and it allowed us to see basic structures within the cells. The last of our procedures was Western blot analysis. Western blot analysis was used to detect the protein expressions levels in the samples. We had our samples and we loaded them into a gel. There are several wells which we put the samples in. We then used a running buffer and attached it to our power source in order to separate the proteins into different bands along the gel. We could then use the gel to use another method similar to the one before, and to transfer it to a membrane so that we can use another machine to read the levels of the protein. Before we did use the machine, though, we did incubate the membranes in 2 antibodies. The primary antibody was consistent of the 3 main proteins found in autophagy that were shown in the diagram before. That way we can detect those levels. Our results showed that cathepsin K knockout did alleviate starvation-induced body weight and liver weight loss. If you see here in the control group, compared to the starved group, there was a significant amount of weight loss. However, between the starvation of the wild type, the control group for cathepsin K and the cathepsin K starved mice, the difference in weight loss was less. So it did less than the amount of weight that was lost between the starvation. This is the results from the H&E staining. We found that cathepsin K knockout prevented liver morphological changes in starved mice. This is our control group of the mice who were not starved, and this was the starved mice. You can see these white circles here, pointed out by the arrows. Those are vacuoles and fat, which represents liver damage due to starvation. However, if you compare this picture to the picture below it, you see that there are much less white spots, showing that there was a lot less liver damage due to the cathepsin K knockout. This is similar results. Instead, this is the oil red O staining showing the lipid accumulation. What happens is that in starvation, there was a lot more accumulation because there weren't nutrients necessary to transport the lipids outside of the cell. So they began to accumulate in clumps. You don't see that with the normal mice who were not starved because they were not nutrition deprived. With the cathepsin K knockout, however, there was also no lipid accumulation, showing that the cathepsin K knockout did help prevent liver damage. These are our Western blot analysis results. We found that cathepsin K knockout rescues liver AKT and AMPK activity changes induced by starvation. AKT and AMPK are important regulators of autophagy, which is why we use these. The important thing to notice here is that between the wild-type control group and the wild-type starvation group, you see that autophagy was enhanced because of the darker bands. The darker bands means that there was more protein expression levels that were higher. So that means that autophagy was more prevalent in those mice. However, between the wild-type starvation group and the cathepsin K starvation group, the bands became much, much lighter, signaling that autophagy was decreased. These are the 3 proteins that we used, that are the proteins from autophagy that were in the previous diagram. And we also found that cathepsin K knockout attenuates activated autophagy in response to starvation in liver, which is similar to the one before. We found that between the control group and the starved group, the bands did become darker, so there was more autophagy. However, between the starvation group and the Captain K starvation group, the bands became much lighter, meaning that autophagy was decreased. Okay, so in summary, we induced starvation on our mice, which would inhibit peak AKT and activate PMK and PK, which would then activate autophagy, which would lead to liver damage. However, with the cathepsin K knockout, we inhibited both AKT and AMPK, which didn't allow for as much liver damage. So in conclusion, this study provided results that support that cathepsin K knockout would be a protective measure in cellular survival after prolonged starvation. Currently, there is a pharmacological inhibitor of cathepsin K in clinical trial 3 for osteoporosis, which is a bone disease. And the benefits of cathepsin K knockout in the liver is attributed to changes in autophagy. Basically, it wouldn't make much sense to give starved people medication when they don't even have money to feed themselves. But since autophagy is in other diseases that don't have to do with starvation, it would allow us to see the way autophagy works and how cathepsin K would help regulate it. I would like to thank Wyoming and NSF EBSCOR for funding my research, and my professor Sri Rayan for allowing us to use his lab, and my mentor Dr. Enon for guiding us throughout the program and teaching us everything we need to know. I'd like to thank the coordinator Lisa for planning everything throughout the program, and Esmeralda, who is both my roommate and my lab partner. I'm also the SRAP counselors and students for making the program fun and entertaining, and my parents were always supportive. Thank you. Any questions?